Okay, sports fans, the time has finally come. We're at our next to last video. The first one here is going to talk about the guys that finally discovered the structure of DNA and what led up to it. The next one that you'll see after this is about the actual structure of DNA in a lot of detail. But right now, let's talk about where we left off. So, the last video we talked about a really famous picture, and it was called Photo 51, and it was taken by Rosalind Franklin. And this video showed us, or this picture showed us how this was a double helix that went down around like that. And thanks to her, she took this picture. It took a huge amount of work to do this. This was not something that was easy to do. She had to stretch out the DNA. She had to fit it in a little glass tube. She had to get it just right to get this picture. Because we're talking small. These are not large things. They're long, but they're minuscule in size as far as from here to here. And she figured out how to do it. So that's why she's a genius. And then, of course, that picture was taken by her associate, Maurice Wilkins, and given to James Watson. And he used this along with a lot of other things. So not just this, but this was the final thing. When he saw this, everything clicked together. I think the famous quote by him is, "The instant I saw the picture, my mouth fell open and my pulse began to race." And he knew what he had. He knew how the whole thing worked. And within just a couple days, I think they worked out the structure of DNA at that point. So who's they? Well, they are Francis Crick and James Watson. And what's their story? Well, here's their quick story. James Watson was American. He studied birds. He was an ornithologist. But he was really interested in genetics and kind of where that was going. Francis Crick was a physicist. We all know about those physicists, but he was a physicist, and he worked on the bomb in World War II and decided that he was never going to work on something like that again, so he quit working as a physicist and began looking at molecular structure and chemistry, biochemistry specifically, and that's where they went. So they both got interested in DNA. They're both working at, I just blanked where. I'll have to look. Hang on just a second. These guys got together at a place called, in Cambridge University, specifically a lab called Cavendish Lab. And in Cavendish Lab, they worked together to put together this model. And they worked for a long time. But, you know, what they were trying to discover is they were trying to discover the actual structure of DNA. What held it together? What was it made of? How was it oriented? How are these different molecules attached to each other? And how did they bond together? That's what they were trying to. So they made a lot of mistakes at first, and they weren't the only ones. Here's a guy that they were kind of racing against. His name's Linus Pauling. I don't know if you can see it. Let me make it bigger. Linus Pauling was in California, I believe, and he was working on the structure of DNA too. And this was not a dumb person. He'd already figured out the structure of some proteins that were making him famous and made him famous forever, actually. But any of you that are familiar with proteins, the alpha helix, he figured it out, the structure of it. But these two guys are racing against him, and there's a lot of events that lead into them beating him, but they all made the same mistake at first. And what was that mistake? Well, here's their early model of it. I think this is actually Pauling's model, but it still illustrates some of the mistakes these guys made. And actually, they called Rosalind Franklin over because she was such a good chemist, and she looked at their model, their original model, which if you look here is not a double helix but is a triple helix. Okay, it also, I know you can't really tell it right now, but this is one of the nitrogenous bases. I'm sorry, they had the nitrogenous bases on the outside instead of the inside, so the bases are on the wrong way, and there's 3 helixes instead of 2, and Rosalind Franklin instantly knew that this arrangement chemically would not work, and so she called she just said, "You guys are idiots, call me when you got something." So she left, and I think that's part of the reason James Watson didn't like her very much, is she was a very blunt person, and he's like, this blunt person who is also a woman has ridiculed me, and I hate her, and so I think that's part of the reason that he ended up taking the picture later. Yeah. I think it's all related. Anyway, so here they are. Those are the 2 big mistakes they made. Triple helix bases on the outside. Rosalind Franklin kind of sets them straight. They're still puttering around. Sometimes they're taken off the project even, sometimes put back on. But finally, Watson, of course, sees the famous picture. He sees this famous picture, and he's like, I think I've got it. And so they start working together, and they figure it out. And here's the picture of them with the famous picture. This is them. So this is Crick, and this is Watson, and this is the model. And if you notice, there's this exact picture in the back of my room, in the back above the aquarium, so you can really look close there. But each one of these little dots on here, I don't know how big we can get this. Let's see if we can try and make it bigger. Each one of these dots is an atom, and each atom's making a molecule, and each molecule is bonded together, and they make this helix, this double helix, and these are the chemical bonds and the molecules, and it's super complicated. And they did a great job of working that out, and that was not an easy thing to do, okay? It's not an easy thing to do, but what did they figure out? This is what they figured out. They figured out the structure, and they figured out it's a double helix. They figured out the bases are bonded in the inside with hydrogen bonds. They made sugar and phosphate backbone, and I'm going to come right back to this, so don't freak out that I'm taking it away, but I want to show you something. So they figured this out very quickly. I mean, they worked on it for years, and then once they saw the picture, it was just a few days they figured it out. And then they quickly wrote up an article, and they put it in a very famous science magazine called Nature, which still exists today, and it's a very, very prestigious journal to be published in. And they wrote a one-page paper, which you'll have the joy of reading. It's a one-page paper about the structure of DNA, and it's one of the most famous scientific papers ever written. You guys will read it. Some of it will be over your head, and that's okay, but you'll get the important stuff, I think. So they publish in Nature, and then they end up winning the Nobel Prize a few years later with Maurice Wilkins, but not with Rosalind Franklin. Okay, so, and that's because she had died. They don't give the award posthumously to dead people. But some people say they don't give the award to women, and back then maybe that's true. So that's the big question. Now, what is the structure of this guy? So let me pull Francis and James out of the way, and let's look at this structure and see what I can show you, because I want you to know these different parts. Okay, so the first thing is to notice it's a double helix. See that? There's one helix. The red one is the second helix going up, and they twist around each other like a spiral staircase. So that's a stairway that goes up. I'm not really showing you very well, but it's a stairway, like a spiral staircase. Now, what is this stuff? Well, this blue is this blue, okay? And if you notice, this blue is this alternating molecules. The first one is a deoxyribose sugar. There's deoxyribose. That's a sugar there. These are the sugars. And then here are the phosphates. And we've seen phosphates before. Phospholipid bilayer, exactly the same type of phosphate, okay? So phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Over here, same thing, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. In the middle, we have our bases. Now, if you remember, these bases have different shapes, and we probably haven't gone over this a lot, but there are purines, which have 2 rings, and pyrimidines, which have 1. And a pyrimidine always bonds with a purine. So you have pyrimidine, purine. Purine, pyrimidine, purine, pyrimidine, purine, pyrimidine, okay? What holds them together? This bond right here is a very strong covalent bond. But these bonds right here are very weak hydrogen bonds, and these break apart very easily. That will become important later. The hydrogen bonds are important. So, what do I need you to know at this point? I need you to know it's a double helix. I need you to know that it's sugar-phosphate backbone. I need you to know it has these nitrogenous bases, and these nitrogenous bases are adenine, thymine, guanine, and cytosine. The adenine bonds with thymine, guanine bonds with cytosine, and that's how it always works. So adenine, thymine, guanine, cytosine, adenine, thymine, guanine, cytosine. And they don't have to be in that order, but that's how they work. You should know that. You should be able to explain that much to me on your sheet. If you can't, then come see me, and we'll talk. Otherwise, that is Watson and Crick, and that is the search for the structure of DNA. But, you know, actually, one thing I forgot to tell you. What's really amazing about these guys, you know, we spend a lot of time talking about the scientific method. These guys didn't do an experiment. They didn't do a single experiment to figure this out. They made models, and they used the laws that everybody else had figured out doing experiments. They applied those laws and turned those laws into the model, the famous model here. That's what they did. They just sat there and did the math, and they sat there and said, wait, you got to have hydrogen bonds, and these two things won't bond because the atoms aren't in Happyville. And they did this until they got it to work, and until they got evidence like this to back up their work, and then they figured it out. And once they figured it out, it clicked, and they knew what they had. So the next video is going to talk more detail about DNA, and it's pretty detailed, but it's worth the time because it's pretty cool. If you have questions, please let me know. Otherwise, peace out, homies.